Okay, hi. So my name is Evan. Some of you already know me. I have a lab where basically the last few years we've been looking at two different issues in autism. One is looking at epigenetics in autism. For most of that, we've been looking at postmortem brain tissue and looking at epigenetic changes. The other thing, which I'll talk about now, is about changes in the microbiome of individuals with autism. About 3 years ago in the first of these conferences, I talked about a mouse model, and he said, one day I'll come back with data about people in Israel and autism, and so this is that day. Talk about that, and also show some mouse data, giving some implications of what we found. So, to remind you, we have in our body the microbiome, which is basically bacteria and other agents found in our body, which we live in symbiosis with them. Most of them do for us different functions, help us metabolize, for example, and other daily functions that we need. And we have a different microbiome and different organs. We have a skin microbiome, oral microbiome, gut microbiome, etc. I also know you don't have a lot of time, so I might speak a little fast, but I just want to help whoever is after me. There is the gut-brain axis. In other words, there's a lot of interest recently in the fact that what happens in a gut can influence what happens in a brain through a few different mechanisms. One of them is that through the metabolites we just talked about, there are many metabolites made in our gut which travel through the blood-brain barrier to our brain and can influence brain function. For example, short-chain fatty acids, which can bind GABA receptors, etc. Another method is through the regulation of the immune system. In other words, of course, our microbiome very much influences inflammatory processes in the body, which also downstream influence brain and function, and also possibly through regulation of neurotransmitters, talk about soon. One other fun fact is if you take all of the neurons in your gut, make them into a ball the size of a cat brain, which means we really do have like another brain in our gut, which can influence perhaps our behavior as well. For this study, we've been using samples which we've been gathering for many years now, a biobank that we made together with Dr. Judy Carmel, who's found here in the audience, and we together have been building a biobank which includes, show here, biological samples, also clinical data and epidemiological data. We have currently around 150 individuals in the biobank, including plasma, serum, saliva, and stool samples. So I'm talking now about the microbiome study that we've done through these samples that we've gathered. And what I'm showing you now is data from 97 individuals with autism here in Israel, 42 controls, showing the different male and female ratios, and they are of average about 8 years of age. This is an average. There is a range here going from about 4 to about 16, 17, etc. The opposite. Okay, so what do we actually do? We take stool samples from individuals. We take out the DNA or extract DNA from these stool samples, and then we sequence them, and through that sequencing information, we can know what bacteria live in those samples. Okay? So the first finding is we look at something called alpha diversity. What is alpha diversity? Let's say, for example, I have 4 different types of bacteria in my body, whatever they may be. I have much more, but let's say there were 4. So that is my alpha diversity. Someone else, if they have 6, they have a higher alpha diversity than me. It doesn't matter how much of the bacteria, it is the amount of species. Okay, that is the alpha diversity. So what this basically shows us, we had a very significant increase in alpha diversity in the autism samples, which basically means they had a more varied microbiome, more types of species found in the samples. Number one. Next way to look at this data is through beta diversity, and this is principal component analysis. Basically, what you're seeing here, again, for those who are not familiar, if you see every dot represents the total microbiome of one individual within the experiment. And let's say these 2 dots, I can't see what I'm doing here. No, okay. So if 2 dots are very close to each other, then that means they have very similar microbiomes. The more they are farther apart, then that means they have very different microbiomes in terms of what species are found there. So if, for example, I was looking at the microbiome of a mouse and a human, I expect to see them at 2 very different places clustering in the analysis. What I do see here is I see more of an overlap. So it's not as if the autism and the control sample is completely different in our microbiome, but what I think you can see is the grand majority of controls, but 90% are clustered over here with about 40% of the autism. And much of the autism is actually found over here. So there is a different overlap, there also seems to be something which is moving a little bit. And that might be some sort of subpopulation, as I might discuss later, going back again to this issue of heterogeneity that we talked about this morning. So when you look at actual things that change, in terms of what species change, right? Just people, we use two different bioinformatical analysis. The first one you hear is called REFSET, and we looked first of all at the level of phylum. So just remember, there's a taxonomy starting with life, domain, kingdom, phylum, class, order, family, genus, species. Say that 10 times. And what we see is that in the autism group, we have an increase of Bacteroidetes. We also, and according to this analysis, also of Proteobacteria and a decrease in Actinobacteria. However, this particular analysis can't take into account other factors such as age and gender, so we took this data and looked again at another bioinformatic tool which can take all of this into account, and so we define covariates for age and gender, and find again that Bacteroidetes increases by a very significant amount in these autism samples. So that's the level of phylum, which is way up here. Again, you can't see what I'm doing, but is up there in the taxonomy table. Sorry, this way. So now going down to genus. Again, genus, for example, if I say Homo sapiens, genus is Homo, okay? So that's where we are in the taxonomy scale. And there we found an increase in autism, again, in both bioinformatic tools and Bacteroides. Bacteroides is again a subgroup, or is a genus that is also under the phylum of Bacteroidetes that we talked about earlier. So this seems to be a pathway of what is increasing in these autism samples. So to summarize what I said up until now, in these autism samples that we have, and we are collecting more samples, we'll be adding on to this data as we go. But what we have until now is we have an increase in alpha diversity, a difference in beta diversity, and an increase in Bacteroidetes and Bacteroides. Now, this is surprising, particularly to myself and other people, because people in the field of microbiome and autism will be very familiar with a paper, one of the first papers published in the field in 2013, where they looked at an autism mouse model and found that Bacteroides, and particularly Bacteroides fragilis, decreased in that autism mouse model. And when in the adult mouse, they gave it back, they treated with Bacteroides fragilis, as that actually attenuated the social deficits that you would see in this autism model. So this paper suggests Bacteroides is good, while ours suggests, well, we see more of it in autism, whatever that means. So here, we actually, very interestingly, for a completely different reason, we were already studying Bacteroides in the lab, and so we got data at the same time, which helps us understand this whole issue. What we had done is we wanted to know the developmental roles of microbiome in behavior. So instead of what people usually do is they take a mouse model and they give to it a particular species during adulthood, well, what if we do it at the very beginning of development? You see, how does that change development to behavior as the mouse progresses or becomes older? So what we did, and this took quite a while to get this to work, is we took very newborn mice, one-time application of a species, and we checked could it integrate or colonize into the mouse, and then how does that affect its behavior and development. So we gave to it actually we gave to them Bacteroidetes fragilis was one of the things we gave, and to both male and female pups, and we gave it to them again immediately after they were born. Then we checked at both 2 weeks of age and 8 weeks of age, which is already adult, in mice if there was colonization. At 2 weeks of age, there was very strong colonization. At 8 weeks of age, was already no difference between controls and treated. So it was colonized for a while, and eventually, as the mouse developed, it goes away. What is interesting, and I don't show here, is that we also tried with other species, like the Lactobacillus, which did not colonize. So not all bacteria colonize, but Bacteroides will. So how did this affect the behavior of these mice? First of all, is we looked at marble bearing and grooming, two indications of repetitive behavior in mouse models, and we found that the treatment with fragilis, Bacteroides fragilis, again, one-time application at birth. When you look at these mice as adults, they had, or after development, they were increasing their marble bearing and increasing their self-grooming, which again is two indications of a repetitive type of behavior. We also then looked at social behavior. And what we saw there is that it did not have major effects on what we call the sociability paradigm, which is a three-chamber test for those who are familiar, how much a mouse will socialize with another mouse within this three-chamber test. However, it very much affected their social novelty, which means that if you were to give it a new mouse compared to an old mouse, not old, but one already knows, familiar and unfamiliar, so usually a mice will want to meet the familiar mouse, so the ones that have been treated with frigidus no longer had any preference between the novel and the unnovel. So again, we had given one time during the developmental period bacteroides, and we see changes in repetitive behavior and social preference. Very important, everything I just told you up until now was male mice. And female mice, no differences. They were colonized. We did not see behavioral differences. Okay? Yes, I really am doing good with time, so that's good. In terms of metabolics, so we actually did metabolic analysis, again, of the mice after development, looking at their metabolome, in this particular case in their serum, to see if there were changes. We saw changes in about 25 different metabolites, which again is interesting because the bacteria very much affects the metabolome. This could be a possible mechanism for its changes in behavior. And what we see is a very sharp decrease. I'm giving an example of one thing that we saw at 25, a very sharp decrease in serotonin levels. I don't know if you can read that there, probably not. But it says serotonin. And so we see about a 60% decrease in the serotonin levels, even though tryptophan levels were the same. I just mentioned that in previous works that we've done in germ-free mice, we've seen that in germ-free mice, they have more serotonin, more tryptophan. So it's very clear that the microbiome is affecting the metabolism of serotonin and tryptophan. However, we wanted to get more insights into what might be going on in the brain, in terms, again, looking at the brain now of 8-week-old mice who had been treated once with bacteroides when they were born. And I'm sure you can't read that, so I just have to tell you, and you have to trust me, that we have a sharp increase in genes that are involved in synaptic function. For example, glutamate receptor activity, structural constituents of synapses, etc., were actually upregulated, which is very interesting, particularly because it's known, or one of the theories is known. One of the theories of a biological role in autism is of dysregulation of the excitatory and inhibitory balance. So maybe there's some involvement here. Again, how do we get from changes in B. fragidus to these changes in gene expression? Are still not 100% clear. In my opinion, probably metabolitic things entering the brain, but still needs to be figured out. So one of the things, before I give a talk, I always like to check one's time in PubMed. Is there something new that I'm missing that I think might be interesting? So this is something published about a week ago. This is actually not dealing with autism, but in depression. Again, a mouse study showing that when you gave bacteroides, including bacteroides fragidus to mice, it had antidepressive effects, and particularly through, depressive-like effects, particularly through the regulation of serotonin. So again, some sort of connection between these two things. It's published about a week ago in Brain Behavior and Immunity. So where are we taking this now? What I've showed you is an update in terms of our microbiome data. We are continuing to gather samples. We have started cytokine analysis through serum samples of all the same individuals, and are planning soon metabolome analysis. The idea is on the same individuals, looking at all these different biological biomarkers that we may be able to correlate, and maybe, for example, look at what metabolites are particularly more present in those individuals with more bacteroides, for example. One last thing before I shut up is I'll go back up to this thing here. What I didn't show you here is actually, when you look at this graph, what I can also do is I can look at where do you have more bacteroides. And what we found is that the bacteroides is found more on the left side, from your point of view, the left side of the sample. In other words, those samples, all these red samples, which is autism, I'm sorry, I didn't mention that before, red is autism samples. So all those red autism samples, which are on the left side, are all the samples that have more bacteroides. So of course, we're also going to go back to the data and hope to find out if there's something else which is defining those individuals or in their metabolome, or even maybe in other data that we have about these families. Thank you all very much, and if you have any questions, I'll be happy to answer.